Welcome to the NOFA Summer Conference, our virtual conference. This is the first format for us, first time we're doing this, so we appreciate everyone's patience and your willingness to join us, and we're so happy to have you here in the first workshop of the series. So my name is Anna Gilbert Muhammad. I'm the Equity Director and Food Access Coordinator for NOFA Mass. I will be hosting you for this workshop. I am joined in the co-pilot seat by Jocelyn Langer. You heard her earlier today in our orientation. She is the incoming executive director, so she's helping me out in the co-pilot position tonight. The name of our workshop tonight is Got Biology, the What's Next After Soil Testing and Amendments with Laura Davis. This is one I've been excited about. We will get into her intro real quickly. We just want to go over a few housekeeping things, and then that will be the last that you will hear of my voice. Then you'll be hearing from Laura. We'd like to thank, first of all, we want to thank our sponsors that helped us put this together. You see on the screen, first of all, our gold sponsors. I will not name them all. Please go to the sponsors that are going to be listed or are listed in our program book. Do business with them. They have greatly and graciously helped to sponsor this conference. In our silver sponsors, you now see that on the screen. Another group of sponsors that have donated to help put this conference on. Again, you should see them in the program book, which I hope you all are enjoying that program book. Again, over the course of the conference and the year, please try and do business with them. And just to wrap up our opening comments, just to pay homage and respect to those who were in the lands that we are all coming from previously, we are presenting, attending, and hosting this workshop from land that was managed and inhabited before European colonization. For example, I am broadcasting to you live here in Springfield, Mass., which was one part of the Pocumtuck territory that spanned from the Enfield border with Massachusetts all the way up through Deerfield. We would like you to take a moment to find your location on this map that you see on your screen now. There is an interactive global map, https://native-line.ca/. I will put that in the chat for you. Feel free to click on that, look at it, and see where you're located. And that way you can see exactly what people were there before the European settlers, who were the actual holders of this land previously. We want to give that respect and homage. So without any further delay, I'd like to introduce to some, and I can honestly say present to others, our presenter tonight, Laura Davis. She is a certified organic vegetable grower at Long Life Farm in Hopkinton, Massachusetts. She's an organic inspector. She is the lead soil technical advisor for NOFA Mass. And finally, she is NOFA Mass's board chair. So at this time, I'd like to go ahead and turn the controls over to Laura. And just to make sure, just to do a quick comms check here, Laura, can you hear me?
I can, thank you.
Okay, very good. So I will give you the ability to share your screen.
So thank you all for joining our first slot of our presentation of our virtual summer conference. I recognized a couple of names that signed in who have seen part of this presentation before. So I hope you're not too bored with some of the chemistry that we're going to go over. This has been a work in progress, and the great thing about this presentation is that it actually has followed a grant that we've been doing at NOFA with some farmers. It's a 3-year program where we've been looking at the soil in terms of carbon proxy testing. We've been looking at mineral balancing, and we've been looking at the biology of those soils and comparing over 3 years. So we've learned a lot over those 3 years. And I hope to share some of the practices that I specifically have incorporated into my farm. So I'm going to talk a little bit just briefly about where I'm farming in Hopkinton, Mass. And the topics that I'm going to cover broadly are really about uncovering plant limitations through various types of testing, both soil testing and plant sap analysis. We'll also talk about addressing those mineral imbalances by analyzing that soil test report. And we'll also talk about adding biology in various ways. So I am farming since 2011. I technically am still a beginning farmer until next February when I will have my 10-year anniversary of Long Life Farm. My husband and I farm on a 2¼ acre certified organic farm. We have been low till for the last 3 to 4 years. The only tilling that we do is with the rotary plow to hill our potatoes. And we grow a variety of diversified vegetables. About 85 farm shares is about 55% of our farm business, and the other half is farmers markets. We do 2 farmers markets in Ashland and Hopkinton. We have a high tunnel that we put up in 2015 where we rotate between tomatoes, eggplants, and peppers to wintergreen spinach, and then following the next year with cucumbers and other squashes. This past year, we put up a low tunnel, which is about 50 by 14, so that we could crop rotate between our tomatoes and cucumbers for the summer. And we also have a seedling hoop house, which is in our backyard. We have been utilizing the Albrecht method since 2011, and I'll talk a little bit about that when we're talking about mineral balancing. And we produce about 30,000 pounds of produce. I have to mention that my very first exposure to farming was volunteering at Many Hands Organic Farm in 2011. And I had left corporate America after 30 years, and I was attending a course that at the time was called the Nutrient Density course. And I met Julie Rawson at that particular meeting, and I ended up working at her farm as a volunteer shareholder in 2011, and really was able to see from April all the way through to December all the things that she did to create fertility and to grow really nutrient dense food. And the following year I started my CSA. So the reason I'm here and the reason I'm farming and the reason I'm with NOFA is because of that first course of Dan Kittredge's. So over the years, over the last 10 years, I've learned a lot about soil fertility. I'm certainly not an agronomist by training. I'm not a chemist, I'm not a biologist, but I certainly do enjoy this part of analyzing soil and creating soils that can create better food for all of us. So as we get started, we're going to talk a little bit about soil testing. And one of the things that's important is that you don't want to guess. You don't want to put compost on your soil if you don't really know if it needs it or not. Generally, we recommend that soil testing is done at least once a year at the same time. I do my soil test every year, end of August, beginning of September. And not all labs do the same type of test. So if you send your soil to UMass Amherst, they will use a test called the Modified Morgan. We recommend Logan Labs as well, because they can give us a lot of identification of micronutrients that we believe are important for not only the crop health, soil health, but also for the quality of the food. So The one thing about these two tests is that you can't, they're not apples and apples. You can't compare one to the other because they're utilizing a completely different chemistry. Some farmers will use both. They'll use UMass Amherst to give them an indication of how much phosphorus their soil has. It tends to be a little bit less aggressive, and so gives a better reading for phosphorus specifically, which we care about in terms of not having so much that we have a pollution risk. The other type of test that we often will do is called a saturated paste test. And that will also show clearer phosphorus levels. But a saturated paste test is going to tell you exactly what's happening in the soil today versus what's in the savings account long term, which is what a regular soil test will tell you. And Anna, you're going to stop me if there's any questions in the chat, right?
Yes, I certainly will.
Okay, thank you. So those are the two types of tests that soil tests that most people will be familiar with. Soil standard test, what's in your savings account, and saturated paste test, what's in your checking account, what's available right now in terms of liquidity, for example. So this is an example of a soil report from my farm. And I actually do like to send it to Advancing Eco-Agriculture, who also sends it on to Logan Lab. So this is in fact a Logan Lab soil report. It just doesn't have their logo on it. The reason that I send it to AEA is because I also work to make sure that I am adding the right amendments. I like to, even though I can do it myself, I can analyze it myself, I like to get another opinion to see if a trained soil agronomist has some other input for me that I'm missing. And so this was the soil report that I took last October, and we'll be talking a little bit about different items on this soil report. We'll be talking about exchange capacity. We'll be talking about organic matter quite a bit. And the thing that I like about this report is that it does give me the amounts or the ideal levels that Advancing Eco-Agriculture recommends for each of these major minerals, as well as these micronutrients at the bottom. This is what a saturated paste report looks like. And oftentimes what we'll do with the saturated paste report is really look at how it compares to the soil test report to see if we can find limitations. I don't generally use this as often, but if there's a problem that I want to uncover, then I would recommend sometimes to do both a saturated paste test and a soil test, standard soil test. So as I look at the standard soil test and what Charles Albrecht, using the Albrecht model of soil balancing, there are certain balances of minerals that are recommended. And the standard soil test in this slide, or in this graph, the purple is my farm's test, and the red line is what Albrecht recommends in terms of ideal soil balancing. And right away you can see that I have some good calcium, but that my potassium is way off. And also my manganese, so forth. So this is the reason we do soil testing, is because we want to balance and remove any limitations that our soil might have that would limit us from producing a good crop. And at the time I did this saturated paste test, it also identified specific limitations that my soil had. Even though my other test, as you can see over here, looked like I had plenty of calcium, on the saturated paste test, it was telling me that that calcium wasn't available. It was locked up. And so these types of tests can be very telling when it comes to things that are limiting the growth of our crops. Another type of test that we've done for our grant that I spoke about earlier is microbial biomass interpretation. Now, this particular test we had done at the University of Maine. And what it will tell you is how your biology is doing. Is it active or not? And we explored this because oftentimes the physical tests that you can take, and Caro Roselle has done many of these carbon epoxy tests to look at aggregates, look at the amount of earthworms, look at the roots in terms of the footage down, and all sorts of different physical characteristics that would tell us how healthy the soil is. This particular test that is done with a standard soil test at UMaine actually gives a number, parts per million, to the microbial biomass in your soil. And so a lower number would mean that you're going to need a lot more biomass incorporated, and they recommend cover crops, compost, and as you get higher on the spectrum to 140 to 300 parts per million, that represents a very high biomass, very strong microbial population. And so they generally, would not recommend 2 inches of compost, but more like a ½ inch of compost. So that's an interesting way to look at testing your soil by checking a box on the University of Maine test that says you want to do microbial biomass interpretation. And it's a minor charge. I think it was $10 or $15 more to get that that measurement. So that was a good way to look at it. Another type of testing that I have been participating in on my farm is leaf sap analysis. And this particular test is on a crop of beets from 2019. I always have trouble with my soil. My soils were very old and tired when I started back in 2011. And I've been continually improving since then, but it's not perfect by any standard of your imagination. So I sent my beet leaves in, and they tested the leaves, and they came back, and I was weak in phosphorus, iron, and boron. And we were able to put together a foliar spray recipe to address my beet limitations, because I wasn't getting any beets to size up. They were just staying straight like little pencils. And I'm happy to report that this particular sap analysis, I haven't sent them back in for testing because I followed some instructions for the foliar test this year on my beets and was able to get some nice round beets, which I'm very happy about. Okay, so let's talk about total exchange capacity, which is one of the measurements that is usually on all soil test reports. The exchange capacity is the soil's capacity to hold on to essential nutrients. And so this picture shows humus, which has the highest level of exchange capacity. Clay in the middle also has a high exchange capacity, and compared to sand, which has a very low exchange capacity. And why is that important? Well, depending upon your soil, I started out with very sandy soil. All these negative, these minus charges on the sand colloid here represent exchange sites where the minerals can attach. And if you don't have a lot of exchange sites, then you can't hold that many minerals. As you get over here to the humus, you then have a lot more exchange sites. And so calcium and potassium and magnesium can attach on, and they're there in terms of being able to exchange nutrients with your plant roots. So this particular exchange capacity, if you have a low exchange capacity, you have to remember that you're going to have to fertilize quite a bit during your season, your growing season, probably up to 3 times, because your sandy soil is not going to hold on to the nutrients as it would if it was a different type of soil with more clay and a higher humus content. That's really important to know when you get your exchange capacity. So an exchange capacity of 12 or higher would be a really good number. My soils tend to have low exchange capacity, around 5 or 6. And so I do need to continually fertilize my plants usually at planting, and then 2 more times during the growing season to make up for this low exchange capacity in my soil. So we're talking about cations and anions. Cations have a positive charge. That would be calcium, magnesium, potassium, sodium, and hydrogen. And anions, which have a negative charge. That would be sulfur and phosphorus and boron. And if you look back at this particular chart, you'll notice nitrogen is also a negative. Here's boron up here, here's sulfur. These are the types of nutrients that we don't generally apply. Well, boron we do sometimes, but something like nitrogen, you would never apply nitrogen in the fall before a winter, because it's just so volatile, it would leach away and not be there the next season. It's not attached to anything on the soil colloid. So these are the types of minerals that you would not add in the wintertime. Okay, so on the spectrum of soil characteristics, on the left-hand side, you have one end of the spectrum, and on the right-hand side, you have another end of the spectrum. And if you think about that picture, humus, clay, sand, high exchange capacity on the left, 60 would be really high. Low exchange capacity would be a 3. These types of exchange capacities often correlate with high organic matter. Low exchange capacities correlate with low organic matter. You tend to have more fertility when the spectrum is on the left versus on the right. And the soil holds more minerals if you have a high exchange capacity. And the soil also holds more water. So that's the importance of the exchange capacity in your soil, and you're going to see that number on your soil test report. The other item that you're going to see is organic matter. And this particular definition is from Vern Gerbinger in Vermont. And he liked to say, plant residues and living microbial mass are the living. Active soil organic matter, or detritus, is the dead, most available to soil microbes. That's really what they are going to be eating. And then stable soil organic matter is often referred to as humus, or the very dead. So the living, the dead, the very dead is organic matter. It's just a different way to think about it. And the target should be at least 5% organic matter. Certainly if you're at 8 to 10 to 12, that's awesome. The more organic matter you have, the better off your fertility. That impact of the organic matter percentage For every 1% of organic matter, you can hold 16,500 gallons of water per acre to a 1-foot depth. And as the organic matter increases, the exchange capacity and your nutrient holding capacity also goes up. The structure and the tilth of your soil improves, and compaction is reduced. It increases the amount of nitrogen that's available to the crop. So for example, if you had 10% organic matter, you get between 10 and 15 pounds of nitrogen per percent in your organic matter. So if you had 100 pounds times 10, you'd have 100 pounds total. So 10% organic matter would give you 100 pounds of nitrogen in the spring for your crop. And that's a good amount. Not quite enough for tomatoes or potatoes, which need around 140 to 180 pounds of nitrogen per acre, but it gets you a long way. You're also going to find greater crop resilience with that higher organic matter and more carbon cycling and sequestration if you have enough photosynthesis. Okay, so how do we build it? How do we build organic matter? And there's a lot of this in here in this section of my presentation, and more as we get into the practical practice of organic matter on our farm. First, cover crops are really critical for building organic matter. Either undersowed, under kale or other crops that are still growing in the fall, or following a crop that's been harvested in August, for example, like garlic. The other way to build organic matter is to either do no tillage or disturb the soil as little as possible. Keeping the soil covered with plants is critical, but if you don't have plants down, then mulch, organic mulches like shredded leaves or hay or straw are really important to keep that soil covered. So plants, natural mulch, or cover crop to keep that soil covered, and cover crop is going to go the furthest in regards to building that organic matter. Increase biology to cycle nutrients. So we're going to talk a little bit more about biology at the end of the presentation, but it's really important to, the biology is critical to cycling the nutrients. They're the little farmers, the little helpers underneath the soil that will help to deliver the nutrients to the roots that your plants need. So biology is really important. Compost, manure, plant residue from prior crops all help to increase the biology. Inoculants. Some of you have heard of seed inoculant that many folks use, a mycorrhiza fungi inoculant that's used on seeds before they're planted in trays or before they're planted in direct seeded into the soil. These actually do help quite a bit to increase the amount of mycorrhiza fungi and other beneficial organisms. And so it's an easy way and a very inexpensive way for you to start adding some biology. And you can add those on seeds at planting, at transplant, or through fertigation through the irrigation system, or just from a watering bucket. And biostimulants really are food for biology. They're things like molasses and seaweed, and you can either use these in your transplant liquid or in fertigation, or even in foliar spray to increase photosynthesis. And the more photosynthesis you can get, the more that plant will be able to utilize and to make the carbohydrates enough for their own growth and for fruiting, but also to supply additional to the root zone in the form of root exudates. When you're reading your soil report, this is a minor thing, but just to keep in mind that sometimes your soil report will be reporting out measurements in parts per million. Sometimes you'll see pounds per acre. The easy way to change parts per million to pounds per acre is to multiply by 2. The pounds per acre is used to calculate most of the major mineral amendments. So if your report does say ppm, it does need to be converted before you start calculating how much your amendments, how much of the amendments you'll be needing to add.
Laura, could we give you a few questions at this
Sure.
point? Okay. There was a question that was asked if you could define what is CFA.
CFA. Was that in on my slide?
I believe so, possibly in the very, very beginning.
CFA, sorry, it's not ringing a bell right away. Oh, CSA.
Okay, okay.
Was it CSA?
It might be. That might be it.
Okay. CSA is community supported agriculture. And the way that we sort of know that term these days is a CSA is a subscription to a farm, and that generally takes place at the beginning of the growing season, and then there's a relationship between the farmer and the shareholder, that there's risk and reward involved in the farming of their vegetables. So generally a CSA, our CSA of 85 to 90 people, they subscribe up front, usually by April, and then starting in June, they will enjoy 20 weeks of vegetables from our farm. So they get to enjoy the harvest. The nice thing about a CSA is that it helps to pay the bills. Most farmers open their CSAs up sometime around Christmas time. And as people join between Christmas and April, when it finally sells out, those dollars are able to be used because they pay for the whole thing up front generally. Those dollars are able to be used on seed orders, on supply orders, and it's a great business model for small farms especially. Thank you for that question.
No worries, no worries. Another question, is there a relationship between the biomass measurement and the Logan Labs organic matter measurement?
Can you tell me that question again?
Sure. Is there a relationship between the biomass measurement and the Logan Labs organic matter measurement?
Not really. Well, the way they would correlate generally would be that the more biomass you have, the higher your organic matter most likely will be. The more biology that you have in the soil, the more that they will help to, not only do they eat organic matter, but it will absolutely help to build that as well. Mm-hmm. And some people often believe that they're helping to build the stable organic matter. And there's others that have a theory that it's the cycling of that organic matter that is the most beneficial. The cycling that the microbes do is the most beneficial to our food production.
Okay, very good.
That's a good question.
Yes, very good. Thank you for that. And then the last question for this rotation, on the leaf sap analysis, why were there 2 rows for each element?
Why was there 2 rows for each element? Okay, let me go back to the report and just look at that real quick again. So one was an old leaf and one was a new leaf. So when they tell you how to do the sampling of the plant, you're supposed to take leaves that are old that were on the outside of the beet, and then I was supposed to take new leaves that were newly grown from the middle of the plant. And they can tell different things about that sap. It's more like a historical view of what minerals that plant had from start to now. So that's a great question. I think I might have answered that. If not, they can ask me a different way. But you'll notice here, young leaves and old leaves.
Okay. And finally, could you repeat the parts per million to parts per acre slide again?
Yeah.
Someone had a request for that.
So the parts per million, this is how that is calculated. The density of 1 part per million of an element in the top 6 inches of soil would weigh 2 pounds if spread over an entire acre. To convert parts per million to pounds per acre, multiply by 2. And often we have to do that type of multiplication first before we can look at how much calcium to add to the soil, for example. Okay, any other questions at this point?
Not at this point. A little bit later on, we'll have some more. Thank you so much.
Thanks. So the next thing that we're going to talk about is base saturation. And base saturation is also on your soil test report, and it's usually shown as a percentage. Let me just go back real quick and show you that. Okay, so I'm talking about this section here that says base saturation. And it gives you percentages, and these will add up to 100% of the base. So if you think about it as all of those exchange sites having something attached to it. In my case, in this field right here, almost 80% of my exchange sites were actually calcium was attached there. Almost 13% were magnesium and 2% were potassium. So if you think about the base saturation of those sites being saturated with these minerals, is the base saturation. And the percentages that are on this soil test, Charles Albrecht had a formula that he researched and recommended for vegetables, and that is what we're going to cover next. So the desired base saturation for growing vegetables is to have the calcium exchange sites to hold up to 68% of them being calcium. Now, depending upon what soil agronomist you are working with, if you work with someone, Advancing Eco-Agriculture definitely has higher numbers than some of these. But they definitely can get you into the these will definitely get you into the ballpark. These are the percentages that you want to work towards. So calcium and magnesium are the major minerals, and potassium is next. Sodium, believe it or not, is needed in soil in some degree. And then hydrogen is, you will often see in the base saturation if those exchange sites are not taken up by calcium or magnesium or potassium. And if that saturation, the base is balanced, the soil pH will naturally land at 6.3 to 6.4. So many of you who have been gardening a long time, you know, you may say I have high pH, low pH. One of the first things that we were taught in this type of work was that we don't have to worry about pH if we balance the major minerals. And it is quite true that if you work towards balancing calcium, magnesium, and potassium, and then after these major minerals are balanced, work on the micronutrients, the pH will naturally land at a perfect pH for vegetables, which is 6.3 to 6.4. And this particular percentage comes from Charles Albrecht's Science for the Ideal Soil for Plant, Animal, and Human Health. So the calcium, magnesium, and potassium are automatically calculated for you on the Logan Lab Soil Report. The ones that are not automatically calculated for you are these: sulfur, phosphorus, boron, manganese, copper, and zinc. And many people do like to test other micronutrients. There are many, many others: cobalt, molybdenum, silica, many others that you don't need to know necessarily right away. But I generally recommend for people to balance the major minerals first, including sulfur, phosphorus, and boron can make a really big difference in especially your beets. And manganese can make a huge difference too in terms of your photosynthesis. So how do we calculate these things? So this is one of the most complicated for people who aren't used to, following these or analyzing their own soil report and figuring out how much to add of a particular amendment. This is the work that I do for NOFA, so if you need help with this, please ask. It's easier for me to do than you to do probably. But the total shortage in pounds. So let's just say that we needed to have 100 pounds, that we were 100 pounds per acre short of calcium. We have a lot of choices when it comes to calcium. We have agricultural lime, we have dolomitic lime, we have gypsum, and there are others, aragonite, and oyster shells, and all sorts of things. But bottom line is we need to know how much calcium is in that amendment before we can calculate how much we're going to put on our soils. And so generally, this is a very simple example, but it tells you exactly what you need to find out. So ag lime, if you were to buy agricultural lime, or high cal lime is sometimes it's called. Excuse me. We would take the percentage of calcium in the product and divide it by the deficit. So our deficit of 100 pounds would get divided by 0.39, which is 39%. And that would give us an amount of 256 pounds of ag lime that we would need per acre. So that's a basic calculation. If you don't have an acre, which most of you are backyard gardeners, if you don't have an acre, you're going to want to figure out what percentage of an acre you have. So let's just say, for example, you have a 2,000 square foot garden. You would divide that 2,000 square feet by 43,560, which is the number of square feet in an acre. And I came up with 0.0459 of an acre times my 256 pounds. And I get 11.75 pounds that I would need for my 2,000 square foot garden. So that's an example. And I don't want to get too much into the calculations, but this is the type of calculation that you would need to decide on amendments for your deficits that are limiting the growth of your vegetables. Okay, and I mentioned this verbally before, excuse me, about nitrogen, organic matter percentage. So if most of my fields are between 4 and 6% organic matter, you would just multiply the percentage organic matter by 10 or 15. The 10 is if it's non-irrigated, 15 is if you have water readily available. If you had used cover crops, you could add another 5 pounds to that number. Or if you had crop residue present, another 5 pounds. And the additional needs that you would sum up would be the total nitrogen that you need to add. Excuse me. And again, if you're using amendments, you're going to want to look at the container, the package, to find out exactly what percentage of the mineral it has. For example, alfalfa meal is 2.6% nitrogen. So you would actually divide by 0.026 in order to come up with the amount that you would need. Okay, so the recommendation is to focus really on your calcium, your magnesium, your potassium, sulfur, and phosphorus first. Those are the major minerals. And then really focus on the biology and cover cropping. And after you get to the point, then you can start looking at your micronutrients as your soil major minerals begin to balance. We also recommend, there's a couple of different ways to apply micronutrients. You can either use a granular type of amendments, or more and more people are moving towards micronutrients in a liquid form that can be sprayed on the leaves directly, because the leaves can take in these nutrients through their stoma. And that really is a great quick way to get these nutrients into your plants while you're building up the levels in your soils, which takes more time. Okay, so we're going to move on to talk about the soil biology specifically. And when we talk about soil biology, what are we talking about? I think that this is just a general definition, but the soil biology relates to organisms within the soil that carry out a wide range of processes that are important for soil health and fertility. They decompose and they recycle soil organic matter. They improve the nutrient availability and soil structure, transmit or prevent diseases, and they degrade pollutants, many of them. So the total number of organisms, species, diversity, and their activity will fluctuate with changes in the soil environment. And that means that if you're using tillage, you're going to have less number, less diversity. If you're using cover crops, you're going to have more. And so if you think about some of the organisms in the biology, we're really talking about, and this area of biology in the soil is such, I mean, we know more about Mars than we do about the biology of the soil, believe it or not. But there's more and more very bright people that are doing research on the biology and helping farmers to understand why it's so important for our quality of life and food. So we're talking about things like earthworms, termites, and other insects. We get smaller in terms of mites, and the microfauna, protozoa, and nematodes, and the microflora such as fungi and bacteria. Those are the ones I think most of us read about in terms of our farming articles, is the fungi and bacteria. I think it would be helpful, and I don't know if this will work properly, but we'll see. I wanted to show a quick 2-minute video. This is from Elaine Ingham's website. And she actually has done a really great job of educating people about biology. And in her, and if she's been at the NOFA conference and done, some great keynotes. But the thing I think is interesting is that she doesn't believe any of these amendments that I've just talked about are necessary if you get the biology right. Now, most people haven't gotten their biology right, but this is a really good 2-minute, I'm not going to play this whole thing, I'm just going to play it to 2 minutes.
What is the soil food web? To understand the soil food web, let's start with the term food web. We all know about the food chain, the animal kingdom on top of which are us humans, right? Well, if we look a little closer, we can see that some members of the food chain don't just eat one thing. That goes for humans too. So the reality is more like a web than a chain. This is the food web. There happens to be a food web in the soil too. This is the living part of the soil made up of insects, earthworms, and much smaller microscopic creatures such as fungi and bacteria. Dr. Elaine Ingham has pioneered research into the microorganisms in the soil over the last 4 decades and has worked with a team of research scientists to understand how they interact with each other and with plants. The soil food web can be thought of as the soil biome. Just as humans have a gut biome responsible for digesting our foods, so too the soil has a biome, which breaks down organic matter and releases nutrients in plant
Laura,
yeah.
You want us to see the video because we're not seeing it.
Oh, I'm sorry.
Groups that make up the soil food web are bacteria, fungi, protozoa, and nematodes. When in balance, these different groups interact with each other and with plants to create abundant ecosystems such as the great forests of the world. Have you ever wondered how forests can be the most productive ecosystems in the world without the need for any fertilizers or pesticides? The answer lies in soil biology. With a healthy biome, the soil can provide plants with all the nutrients they need. And
I've got to change the screen share. I thought I could just do that by moving to a different part of my screen.
Is it a YouTube video, Laura?
It's actually on the Soil Food Web website.
I find if you stop sharing and then share again and choose the new screen, it will work.
That's what I was trying to do here. Let's see. Are you still seeing my PowerPoint?
No, not at this point.
I stopped sharing. Anna, it's telling me host disabled attendee screen sharing.
Okay. Try it now.
Okay, there we go. Okay, I'm gonna do this again because it's a exciting. Can you see it now?
what is the soil food web? To
Yes,
understand the
we
soil
can see it.
Let's start with the term food web. We all know about the food chain, the animal kingdom on top of which are us humans, right? Well, if we look a little closer, we can see that some members of the food chain don't just eat one thing. That goes for humans too. So the reality is more like a web than a chain. This is the food web. There happens to be a food web in the soil too. This is the living part of the soil made up of insects, earthworms, and much smaller microscopic creatures such as fungi and bacteria. Dr. Elaine Ingham has pioneered research into the microorganisms in the soil over the last 4 decades and has worked with a team of research scientists to understand how they interact with each other and with plants. The soil food web can be thought of as the soil biome. Just as humans have a gut biome responsible for digesting our foods, so too the soil has a biome, which breaks down organic matter and releases nutrients in plant-available form. This is how nature has been feeding plants for billions of years. The major groups that make up the soil food web are bacteria, fungi, protozoa, and nematodes. When in balance, these different groups interact with each other and with plants to create abundant ecosystems such as the great forests of the world. Have you ever wondered how forests can be the most productive ecosystems in the world without the need for any fertilizers or pesticides? The answer lies in soil biology. With a healthy biome, The soil can provide plants with all the nutrients they need, and with a number of other benefits, such as protection from pests and diseases, protection from drought and from flooding. The soil food web is, essentially, nature's operating system. Unfortunately, we humans have disturbed the soil food web in almost all of the soils that we manage, causing it to become unbalanced. As a result, the plants we grow struggle. Plowing is the major cause of the problem, as it destroys the larger microorganisms, such as fungi and protozoa, leaving the soil food web out of balance. This results in a system breakdown. Nutrients are no longer made available to plants, and protection from diseases is compromised. Before the Industrial Revolution, humans would plow using oxen or a bull, which provided around 3 or 4 horsepower. Modern tractors can yield 400 horsepower or more. So far more damage is done to the soil biome by modern machinery. The use of chemicals has compounded the problem. The good news is that we can restore the soil food web to most soils within just a few months. This results in a number of benefits, both for farmers.
Okay, I'm going to stop it there since you don't need to see the rest of it. And I'm just going to go back to my PowerPoint. Okay, there we go. So I wanted you to see that, that movie because I think it really beautifully explains the biology that we talk about and the ever growing body of information that scientists are discovering about the biology. Interestingly enough, that video did compare our gut microbiome to the soil biology, and many of the very same bacteria that we have in our body are also in the soil, found in the soil naturally. So we are connected in more ways than one, for sure. And I know that Tim LaSalle, our keynote speaker, will also be speaking about farm inputs and trying to make our farm systems one where we don't need to bring inputs in. I think that one of the things that I've learned over the years is that sometimes when you're starting with really tired soils, because of prior abuse to that soil, either from tillage or chemicals or so forth, balancing the minerals and using cover crops and adding biology can often help you jumpstart your system. So the next part of the presentation is really going to be talking about how I've incorporated some of these into my farming system at Long Life Farm. So we'll talk a little bit about seed inoculant, a fall primer, transplant drench, natural mulches, cover crop, and foliar spread. Any questions at this moment?
Yes, there are a few. From last time, there was a question concerning tilth and what that is exactly.
So tilth, I wish Carol was here to answer that question, but it's kind of the feeling or the structure of the soil, that the way it holds together with aggregates and so forth.
Mm-hmm. Now, in a past slide, you showed your report, I believe that was, and it showed 70–75% calcium. Are there differing recommendations? And there was it looked like it may have been short of calcium, but had 79% base saturation, which is too high. So there was a question coming in about that difference.
For that particular soil, actually, depending upon where the other minerals are, as I recall, I was going to add no calcium at all to my soils. And when I got a professional opinion from AEA, they believed that I really needed to add some gypsum. And this, not only because one of my fields was short on calcium, but also because I needed sulfur. So that particular amendment, just gypsum has both calcium and sulfur. So you can just add one when you need both of those amendments. But I think that generally Calcium is the leader. It's the one that you want to be high, and you want to fix it first above anything else if it's not at least above 68% or so. Okay.
Okay. And this is in reference to adding amendments for permanent beds. What's the best way to apply amendments?
Well, that's probably, I'm thinking they're not going to be incorporating it because they're not tilling there. In fact, I don't incorporate any of my amendments. They're all on top because I'm a no-till farm. And it's amazing how they make their way down into the soil from rain, worms. I'll often put them underneath the mulch that I'm using. So at least they're covered up, but I don't incorporate it. So I imagine if you have a permanent or perennial bed system, you're going to be doing the same thing.
Okay. And just a follow-up question, is there a particular, when's the best time to do that in correlation to the best time to actually get a soil test?
Well, I like to do a soil test in the late August to September. So then I know exactly what I'm going to need to add in the fall. Some of the amendments are best added in the fall so that they can start to break down and incorporate themselves into the soil. Many people seem to not get around to do their soil test until spring. And that's okay too, if that's, when you do it. Just whenever you do it the same time every year so that you can compare. I don't add any nitrogen or potassium in the fall, but I do add everything else that my soil test says I need in the fall. And then in the spring, I add something like an organic nitrogen, like a feather meal or a alfalfa meal or something like that for a nitrogen amendment. But no matter when you do it, just do it at the same time every year. Okay, I'm going to just sort of move on to just practices and some pictures of things that I've incorporated over the years onto my farm. As I mentioned, we've been no-till. This is our 4th season. So in preparation for winter, I think is, the way I wanted to start this conversation was mid-August to mid-September. Generally, I'm broadcasting cover crops, and it's not on every part of the farm. It's on any place I can get the cover crop where I don't have cash crops still in the bed. So I have winter markets, so I'm growing really the last harvest from the field is generally the first week in December, and then I'm growing in the greenhouse. So those plants that are still in the field in December, like carrots, like some turnips, some radishes, I don't put cover crops on those beds because they're still in cash crops until late in the season. For those particular beds, I actually mulch over the winter. So it's either shredded leaves, straw, or hay. So those beds get mulch that are planted late. I also use a fall primer, and that I spray that with my backpack sprayer onto the leaves or the straw or the cover crop where I have it. And my fall primer is a mixture of 3 different products, which includes Spectrum, which is the inoculant. It's got some beneficial microbes in that product. This is advice that I received from John Kempf on one of his podcasts, which I thought was really interesting. He has compared using this fall primer on soils that have no cover crop and on soils that have cover crop. So in other words, he's used the exact same recipe on just the soil, and then the exact same recipe on a field that also had cover crops. So the cover crops in this instance were sprayed. And he felt that the soil that had no cover crops on it was dramatically improved over the winter time. And certainly the one that already had the cover crops on it was even exponentially improved. So this is something that I've done the last couple years, which this year I really was very happy with the way the soil felt in the spring. It was a particularly problematic area of my field. Has never been successful. And finally this year I felt like when I was planting, it was loose. It was, you know, the soil was a much higher quality, and the plants are doing well there this year too. So I'm definitely going to continue to repeat this process of the fall primer. This is just an example of some cover cropping that you can do. This cover crop on the left was a winter kill cover crop around October 11th. So this got on there sometime in August. This one in the middle is kale and Brussels sprouts that has cover crop in the walking paths and underneath the plants themselves. And you'll notice, you know, we haven't harvested the Brussels and we haven't harvested all the kale yet in that picture. This was late October and November, before a frost anyway. And then the one on the right was garlic. It used to be an old garlic patch that we seeded cover crops, peas, vetch, and oats in, and prepared it for the next year. This is the field that we mulched with shredded leaves. We have a relationship with a landscaper who makes sure that it has no grass in it, but it is shredded up nicely so that they don't blow away, which is awesome. You'll see in this picture right here. So this is the bed before the leaves went on, and these are the beds with the leaves on. And this is the leaf cover that got the fall primer. Okay, so spring bed prep. Around mid-March, I go out to all of the beds where I'm going to plant summer crops, Memorial Day or later. So any beds I have planned for tomatoes, peppers, eggplants, cantaloupes, winter squash, summer squash, they all get a spring cover crop of peas and oats. I find if I don't get them in mid-March, they don't grow tall enough to really make a difference. So I'm sometimes out there just after snow, just raking those seeds in to make sure that they get incorporated. And so around 1 week before Memorial Day, we mow that cover crop, and then we plant directly into the stubble. I've seen the French market gardener do this technique as well. He actually covers the stubble with soil first and then solarizes it with a tarp, but I simply mowed it and planted directly into the stubble. We use, on all transplants, we use a drench, which is the same exact recipe as the fall primer. Rejuvenate, Sea Shield, and Spectrum. That's the inoculant. And then we mulch all of the plants with hay. In the walkways, we use a Hopkinton Pro Black Weed Guard fabric. And this is a picture of what that looks like. The tomatoes, these were just planted in this picture. You can't see the stubble of the cover crop because it's completely covered now with the hay mulch and the Hopkinton Pro Weed Guard, which we have used for over 5 years and have not replaced. It gets rolled up on a spool and we use it over and over, and it's great stuff. And over here are some peppers that we mulched and also have the weed guard in between. Okay, so for early crops, those beds that we have leaves on in the winter need a little bit different spring prep. We rake the shredded leaves off of the bed into the pathway. We broad fork the bed with my favorite broad fork from Meadow Creature. We compost. We put, about a ½ inch of compost down on the beds. And we use that same mixture for our drench at transplant, Rejuvenate Sea Shield Spectrum. We transplant those plants into the bed. And then if the leaves are not thick enough to prevent weeds in the pathways, we'll put down the Coverton Weed Guard Pro. And generally I have found the leaves are already breaking down from over the winter. So we often do need to put the weed guard down to make sure we don't have a lot of weeds. This is an example of an early planting of onions. And we put the Coverton Pro weed guard in there. Okay, so last, I'm just going to bring back this leaf sap analysis and talk about the foliar spray, the recipe that we use for our beets. So we use some liquid phosphorus. We use some, that's about an ounce of phosphorus, about an ounce of iron, and about an ounce of boron into that Sea Shield. Which is similar to a fish product, except it's made with shellfish instead of the fermented fish, like you might see with some of the other products that are used. And so that this particular foliar spray, as I mentioned, was quite successful, and all of these products were liquid. And even though I do add some of these products as granular micronutrients, specifically referring to iron and boron, with a foliar spray, you can get a faster impact to the plant right then than you can by using the amendments in the soil. So it's really really a great way to address some limitations that you have during that season. Okay, so I'm almost to the end. I'm at the end of my presentation. I'm happy to take any other questions that people have.
Okay, I have a few more questions, and then we're going to open up and take some questions from the audience as well. There was a question concerning the order after peas, oats, and vetch, which you may do after garlic. Does that set the stage for a different crop in the spring of cover crop?
It does, because that particular bed will be, if I didn't use vetch, that would be a winter kill bed. And I could do anything early in that bed. If I use vetch, it's definitely going to be a summer crop, because the vetch comes back. It's not a winter kill cover crop. And sometimes you want that. I mean, you could do that. You could use vetch or instead of the March seeded oats and peas.
Okay. And in reference back again to peas and oats, does mowing kill those two cover crops?
Not necessarily. The only reason that it was killed was because I mulched on top of it. Mm-hmm. But I have found that a wayward pea or a wayward oat is no problem. Okay. You know, if they grow up through that, I just leave them go, or we weed them out. They're not a big deal, because I don't get that many that grow up through the mulch.
Okay. I want to go to Anita. I believe she had a question. And Anita, if you want to take yourself off of mute to ask this question directly, because I think it's a good question that might come better if I think you were asking Carol a question, and Carol was going to let Laura tackle the John Kemp trial question. So if you can just ask that question concerning that part, I think it's a good one. Mm-hmm.
Sure. I guess I was just wondering whether he also did a trial. In doing that trial, did he also have one that one field that was just cover crops without the amendments as a comparison, as a.
I don't think he did. I don't think he did in that particular instant. I just found it really, the reason I wanted to try it was because he mentioned that the soil was as good as doing cover crops without the extra foliar spray on it. So in other words, what he was saying was that if you didn't get your cover crop down, you could do the fall primer and, you know, still have a leg up. If you were lucky enough to already have your cover crop down and also did the fall primer, it was just added gravy.
Okay. We have a few minutes left in this wonderful presentation. We still can take a few questions. If you have a question that you have not placed in the chat, if you want to take yourself off of mute and ask Laura directly, please do so.
Hi.
Hey, Monique.
Hi there. My question is about the inoculant, and when you're talking about the beneficial microbes, do you know what microbes are in that spectrum, it's called?
Without me going to get the envelope, I couldn't tell you exactly which ones those are. I know there are 5 different beneficial bacteria in that particular product. But they definitely, you can definitely see it on their website. They have a product information sheet, and it will list out all of those microorganisms that are in it. Even if I had it in front of me, I probably wouldn't be able to pronounce them.
Have you ever tried a compost tea, inoculating with a compost tea?
I haven't. I'm intrigued by what Elaine Ingham does. She recommends a compost tea if you have the right bacteria in your compost to begin with. My compost pile is a weed pile. There's no manure or anything in there, so it's not as biologically active as other compost could be. But certainly making compost tea would be a great thing to do if you have some good a good compost to make it with.
And Laura, this was an earlier question, but I thought it would be good to save this to the end. You mentioned Albrecht in the beginning. Do you have any particular favorite resources of his that you can give out to folks?
I actually wouldn't recommend reading any of his books, only because they're like about this thick and they will definitely put you to sleep. The Albrecht papers, there's volume 1 and volume 2. They're pretty dry. You know, the best book I would recommend really is The Intelligent Gardener, because The Intelligent Gardener, it puts this whole Albrecht method into layman's terms. I went to the nutrient density class twice because it was so complicated. I figured you need to go back and go again and learn and have a review. And it wasn't until I read The Intelligent Gardener that it all sort of clicked. It's a great book. It's an easy read, and I highly recommend it because it'll really go through all of this mineral balancing for you. Make it very understandable.
And we
Charles Albrecht, by the way, was a scientist in the 1920s and 30s in Missouri, and he did a lot of research, and you know, he was way ahead of his time, and universities never took on his theories and research. Unfortunately, they went the other way, to the dark side.
Okay. We've got 5 minutes left. If there are any other questions out there. Laura, any other closing comments?
No, I really do appreciate everybody attending tonight, and taking advantage of this great opportunity to see so many NOFA Summer Conference workshops. I know if you're anything like me, you always had trouble deciding which one to go to because you wanted to go to many of them that happened at the same time. Well, now you'll be able to see all of those other ones that happen on seed saving during this time once you get the recording. So it'll be great.
Anna, there was a question earlier about people getting the slides, and I'm not sure if you've addressed that. I don't know if people can email Laura to get copies of the slides or what.
Laura, what is the best way if they'd like to get this presentation or to contact you directly concerning soil technical assistance?
Okay. Laura Davis, it's all one word, at nofamass.org.
And we do have one last question. I think we could probably close out on this. What's the minimum acreage necessary for an organic farm in your estimation?
There's no minimum. There's no minimum acreage. I inspect about 70 operations for Bay State Organic every year. And I've seen farms that are, 0.2, 0.3 acres. There have been some homesteaders who've decided to certify their homesteads as well, even though they, are 40,000 square feet. So it really, you don't have to have large acreage. In fact, the majority of the farms that are certified are small holdings. But if you have questions about certifying, I'm happy to help with that as well.
Okay. All right,
Laura, I just wanted to tell you I was out at Ruth Green's place today. She's one of the folks in our technical assistant grant that Laura and I have been helping for the past 3 years, and she reported to me that this year her vegetables have hit a whole new level. They're just so healthy, and I saw them; they were so vibrant. And she was saying she's been gardening there for like 20 years, and she said she thought that the mineral balancing work that she's been doing with you and the trace minerals that she's been adding with your help was what made the difference. So,
oh, good. That's great. Yeah. Good to hear.
Good feedback from her.
Awesome.
And if you do want more or additional information on our soil technical assistance program, you can go to nofamass.org and go to that link or to that webpage, and you'll see more detailed information about our program.
We're very fortunate. We have about, including Anna and Caro and I and Allison and Jane and Julie and who did I miss? We have a soil tech group. Andrew, we have a soil tech group. We meet once a month to talk about all things soil. So we're bringing all of the knowledge and the cutting edge to our membership. We're pretty excited about that.
Well, unfortunately, we have come to the end of this wonderful workshop. This is something I could probably sit and listen to a great deal. I'd like to thank everyone for joining us, for your patience in this opening session of our summer conference, particularly with some of our technical glitches. Wednesday, we'll have a smoother run. And we hope that you come back and join us for another round of wonderful workshops Wednesday night, starting at 5 p.m. Please utilize that wonderful program book to go through, to look at our vendors and to look at the other workshop selections. So really on behalf of Laura, myself, Carol, and Jocelyn, we're signing off for tonight. Look for the recordings to come later on in the month. So have a great night, everyone, and thank you so much.
Good night.
Good night.
Thanks, Anna and Laura and everybody.
See you Wednesday.
Great presentation.